Anyway, today it's my pleasure to introduce our special guest, Dr. Seong Han. She's currently a postdoc at the University of Munster, working with Wolfgang Linke. I hope I pronounced his last name right. She studies single myofibril work. Previously, before going to Munster, she was a postdoc in the HPL under the supervision of Walter Herzog, who she also she completed her PhD with in 2020, where she studied the individual contribution of quadriceps muscles to patellar tracking and pressure in the patellofemoral joint. Prior to that, she completed an undergraduate degree at Yonsei University in South Korea in physical education. So I kind of roped her into to doing this. I heard she was coming for the muscle symposium, and then we immediately had an opening on this date conveniently, and she graciously agreed to do it. I think it's one of these, like, she did the same to other people, so then she feels like there's some karmic retribution or something. So I'll turn it over to Seong now, who will be talking to us about the contribution of Titin to passive force in single myofibrils.
Thank you, Michael, for the introduction. Can you hear me well? Yes. Okay. So yeah, I'm gonna talk about Titin, but before that, let me just quickly go my introduction that I prepared quickly. So as Michael briefly mentioned, that I did my PhD in Canada for 5 years, and then I also did my first round of postdoc for 1.5 years. And that's when I started learning or studying about single myofibrils, and then I thought that's field for myself. So, and then since I wanted to learn something different, when I was doing my postdoc with Walter and single myofibrils, I primarily used cardiac muscle when I was here, while all group members were working on skeletal muscle. And now I decided to move to another lab that I could work on single myofibrils, but now I'm primarily using skeletal muscle, studying Titin, whereas all other group members in Germany, they primarily work on cardiac muscle. So I don't know, I don't know what kind of balance I'm doing now, but that's the word that I'm working on. And so today I'm going to briefly talk about what I've been doing in Germany, and it'll be about single myofibrils. So starting from the familiar figures that we've been using a lot. So this is a skeletal muscle, and as many of you already know, skeletal muscle has a structure, and as you go down to smaller scale, you have less passive structure that stays at the level. And as I briefly mentioned, I am primarily interested in this single myofibril, where you can go the smallest level, still you can see it, and then you can capture the video. If I take this red part that I colored here and then put them under the microscope, what you see is, I hope you can see it better, or could you turn off the light if it is possible? Thank you. Yes, that's a lot better. So you can see that in this photo, small structure is repeated and then somehow it's attached in a row. So first, this thin line of the image to the next thin line, it corresponds to the Z disc in a sarcomere. And also in this thick dark area corresponds to where you find myosin filaments. And also there's an empty space in this area between thin and the dark thick band in this image. This is where you find actin filaments. So having a sarcomere from Z disc to the next Z disc, actin and myosin, and this is where you find all the classic theory about how muscle contracts and the force generation. But to me now, since I work on Titin, this schematic needs to be added one more component that I'm gonna talk about Titin. So Titin here, is a passive structure that connects from the Z disc, and that goes to the I band region here, and then it goes into these thick filaments here. And actually it goes further down there till the middle of the M band. And this titin structure repeats multiple times, so that way titin stabilizes the thick filaments within the sarcomere. I'm gonna talk about mostly the parts that can be stretched. So let me just cover this part, and then let me focus on the spring-like parts of the titin. So as you can see here, titin is also one of the key components in a sarcomere. It's been also studied heavily related to how titin contributes to passive force and how titin contributes to active force. And I believe one of the hot topic in the HPL is that how titin is engaged to active force generation related to force enhancement and force depression, which cannot be really explained by classic theory of cross-bridge theory. In order to quantify the contribution of titin, what can be done directly is that we measure the force, passive force and active force, before and after we make a small changes in titin. So the question is, how do we make a changes? And there have been clever ways to make a small changes in titin, and let me briefly introduce about how it was done. So the first way in early period of this research, what they did is they exposed the muscle fiber bundles and exposed it to ionizing radiation in low doses. So after they measured the passive and active force before they put the muscle in there, and then they shoot the beam to the muscle, try to break this titin here, hoping that the titin is not connected anymore. And then they re-measured the passive and active force. So that way they can compare the force before and after the intervention. So another way to do is to use chemical degradation. And this work has been done in HPL too. So what they do is they, again, the same manner. So they measure the passive and active force before the treatment, but then after they measured, they put the whole muscle sample to chemicals, including something like trypsin or a high concentration of salt, hoping that this titin can be dissolved by those chemicals. And then you grab the sample again and then re-measure so that you can also compare the force before and after treatment. Another way to do here is to use the animal model that has been genetically modified. So in this case, when the animal is genetically modified and all after biological procedure, what we get is 3 genotypes: wild type, heterozygous, and homozygous. Usually in heterozygous, they do not have any titin part that's affected by this genetically modified. But if you go further heterozygous and further go homozygous, what's done is that the animal has a specific part or region in their titin removed or replaced to the others. And what they do is then compare the force values that was measured from wild type, heterozygous, and homozygous. So that way they can compare the force with the related to changes in this titin. So there have been a few ways to make small changes in titin to quantify the contribution of titin, but there have been some limitations related to this methodology. So starting from these first two, since this ionizing radiation or using the chemicals to make a small changes in titin, it is actually not titin specific. It's not you shoot the beam to the titin. It's more about you grab the whole muscle bundle and then the whole muscle goes through the intervention either with the radiation or chemical degradation. So which means other protein structure in skeletal muscle or cardiac muscle can still be affected. So even if you find decreased level of force, after this treatment, you cannot be really confident to say that decrease in force could be related to titin degradation or the damage from the other structure as well. Also related to this genetically modified animal model, One thing that we need to be careful when we use the animal model with the genetic modification is that the animals with modification, especially homozygous, where the animals born with the reduced amount of titin, their properties needs to be similar to wild type that do not have any genetical modification. But in their research, what they also reported is that whenever the animal goes through the genetic modification, they seem to have secondary disease phenotypes, such as muscle atrophy, like the smaller size compared to the wild type animal. So they tend to find decreased force in homozygous animal model compared to wild type, but we are still unsure if it is because of the titin modulation or because of the disease-related phenotype. So recently, a new animal model has been established which can at least overcome these two limitations. So this is again also genetically modified animal, but this time they have a specific site in their titin that can be cleaved. And this cleavage site is activated, only can be activated by a protease from tobacco etch virus. So it's not the protease that your body can generate naturally. So the protease needs to be gone from the outside, and then this cleavage site can be activated, then the titin is cleaved. So before the protease approach to the muscle, it stays as normal as wild type. Also, this model has a strength because the number or the amount of this cleavage site can be determined by the genotype of the animal. So starting from the wild type animal do not have any cleavage site in their titin. So even if we introduce the protease from tobacco etch virus or TEV to break down the protein of the titin, you will never see any difference in this model because there's no specific site to be activated. Moving on to the next genotype, which is the heterozygous. The animal now this time has a 50% of titin cleavage sites, meaning that 50% of titin has this red dot, whereas the other part of titin do not have cleavage site at all. So when the protease is introduced to the muscle, what will likely happen is this cleavage site is activated, and therefore titin gets cleaved or cut, and this spring-like structure, if it is overly stretched, it tends to go back to or recoil back to the set disc. Whereas the other part of titin that did not have any specific site to be cleaved, it stays as it is. Last model that I need to introduce is that homozygous, where now all titin has a cleavage site. So as soon as the protease is introduced to the muscle, this cleavage site is activated, and therefore all titin will be cleaved. So this is the animal model that's been available since 2020. And so one strength of this model is that we can compare then the force before and after the titin cleavage using the protease, but also across the genotypes. So that way we can provide concrete evidence on titin's contribution to force. As soon as this model was available, there was one attempt try to quantify titin's contribution to passive and active force, but let me just focus on the passive force for this talk. So starting from the wild type, so this graph y-axis shows the normalized force and as a function of the sarcomere length in the average. The gray curve is the muscle force before the treatment. And then after the treatment, which is shown as black line here, they did not see any force drop, which was expected because in wild type, there's no cleavage at all. So even though protease is applied, nothing changed. What about in heterozygous, where titin has 50% of cleavage? So again, here, the same graph, now this pale blue curve is the passive force before the protease is introduced. But after the Titin was cleaved, now shown is this dark blue, they found force decrease by 18 to 28%, depending on the sarcomere length they measured. So obviously, when cleaving Titin 50%, they saw a decrease in passive force. And they further found bigger force drop when they use a homozygous cleaving 100% of Titin, ranging from 50 to 55%. So they confirmed that, okay, Titin cleavage induces decrease in the passive force, and also we see bigger force drop when we cleave more Titin. So I thought, okay, that result kind of makes sense because the number makes sense. But then one thing that I was also curious is that because in the literature, Titin is thought to contribute to passive force up to 90%. But if it is really true, and if they really cleave the Titin 100%, wouldn't this decrease the force by 50% too small? I expected to see a lot bigger force drop in their results. So I went back to their paper and then figure out maybe potential one reason why I tend to find the smaller force drop compared to what I expected. In the method, what they said is that they use they measure the force from the bundles of muscle fiber, where this structure, 5 to 10, I think, fibers are attached together. And in this level, even though these whole bundles are permeabilized and skinned, there are still many passive structures remaining, including membranes surrounding the fiber, and then also connective tissue among the fibers. So I thought, okay, if there are other passive structures remaining, even if the Titin is cut, there are still other possibilities, potential contribution from other passive structure. So this is where we got our research question. So if we then repeat this exactly the same experiments, but now moving down to myofibril, where we can now remove all potential contribution from the untargeted passive structure, what will you see? So that has become our purpose of this study. So we aimed at re-examining the contribution of Titin to passive force in a single myofibrils. So before we started, we thought about what we could see. So first, we thought we could at least confirm the previous findings, where we find bigger force drops when we cleave more Titin. So that's going to be the first hypothesis. And also second, we expect to see more dramatic force drop in single myofibrils compared to what was found when the result was found in fiber bundles. So the methodology, we used some mice animal, and we also used psoas muscle for now. As a preliminary result, I only have a couple animals per genotype, but I'm trying to get more as soon as possible now. So this is the setup that I built thanks to huge support from Walter's group. So again, here, so this is the single myofibril mounted, and the left end, it is fixed to the needle, so I can adjust the sarcomere length to the desired length. It's the right-hand side is attached to one side of the cantilever, and this cantilever is a reference one. So when I stretch the myofibril, the cantilever only this side move relative to this one. So knowing the displacement of the cantilever and then knowing the stiffness of the cantilever, we can then calculate the force of this myofibril. So we measure the passive force at multiple length range, covering from 2.0 to 3.4, which is what they did in the previous study. And then, so first we measure the passive force at 8 different length range by increasing 0.2 microns. And then we shorten it back to the select length and then incubate with the protease from the tobacco H virus. And then after we incubate it is done, then we rinse it with the relaxing solution and re-measure the passive force again. The key outcome for this study is going to be passive stress, which is the force in nanonewton divided by the cross-sectional area of the myofibrils. So this is going to be my results slide. Let me briefly explain first. So I put the same format, which is 3 different genotypes. And this is before Titin cleavage. And corresponding force result will be shown below. Each graph has the same axis information. So first, y-axis shows stress as a function of sarcomere length. This black dark circle is the passive force that I observed before Titin cleavage again. One thing that I wanted to address here is that the passive force for each genotype seems to be compatible. After Titin cleavage, the curve is shown as a red dot here. So starting from the wild type, as we expected, because there's no Titin cleavage site at all, after Titin cleavage, we didn't see any huge changes in the passive force compared to before Titin cleavage. Moving on to the heterozygous, where we cut 50% of Titin. As we stretch the myofibril, we tend to find force decrease at each length range that we measured, and the range was it depended on the sarcomere length we measured, ranging from 8 to 66% compared to the before Titin cleavage. And we tend to find a bigger force drop when we use the homozygous, where we cut 100% of Titin, ranging from 34 to 89%. So in this slide, what we can confirm is that when we cleave more Titin, we tend to find the bigger force drop compared to before the Titin is cleaved. So now let's compare these values to the previous study that used fiber bundles. So on the upper level here, this is from ours, and this is the paper that used the bundles of fiber. So I wanted to confirm that the changes that we observed using our preliminary result is actually bigger than what was found in previous study using the fiber bundles, where still there are many passive structures left. So this also confirms our second hypothesis. So yeah, I mean, I got 2 hypotheses relatively okay. So I thought I was going to be happy, but actually I was not happy with this, or I should say I was not fully satisfied with these results, because let me go back to the results that I had here. In the literature, Titin was primary key elements contributing to the passive force. So my hunch before I start the experiment was that when Titin was cleaved 50%, I actually expected to see this blue line here, which shows the force drop about by 50%, but each length range. The same as here when homozygous was used, or the 100% of Titin was cleaved, I expected to see force drop at each length range, but obviously that was not what I found here. So I got a fundamental question to myself: did I actually cleave all Titin given the muscle sample? So I wanted to be very, very careful with this. So I searched the fastest way and reliable way to prove this answer. And then one way to do was running electrophoresis or Titin drills. So I can identify the proteins information in there. So let me just quickly explain what I found. So 3 genotypes are shown here again, and this is what it looks like whenever I run electrophoresis with the muscles, and then I'll go through with you guys. So here that I labeled here as a Titin. Titin is actually a really big size of protein. So when you load the gels, when you load the sample through the gels, almost the top band is where we find the Titin. And this is when Titin is full length from the Z disc, and it goes all the way down here in the M band in the middle part. And since this is before Titin cleavage, we should be able to see this top band from across all three genotypes. And then what would happen if we cleave Titin in here? So starting from the wild type again, where it does not have any cleavage sites. Since there's no cut cleave or cleaved Titin, what we should expect to see is the full size of Titin stays as it was and as similar as before we cleaved Titin. And when I actually they analyzed the data set, there was no difference between this before Titin cleavage and after Titin cleavage using wild type. Moving on to the heterozygous, where we have 50% Titin cleavage after the protease worked. So what I expect to see was that this top band, which Titin has full size, I wanted to see, or expect to see, this intensity goes a little lower, or lower number of Titin molecules in top band. But since Titin was cleaved, which means that the full length of Titin has been split into two, so smaller size, so likely it goes, the two fragments of Titin will go a little lower band, which is what I found. I hope it is clear enough to see, but the intensity from compared to this one, we kind of find this is reduced compared to before Titin cleavage, but we tend to find a higher intensity in lower size of proteins. So which means that Titin has become somehow smaller after Titin was cleavage. But since this is a 50%, this is why we still see some of them in here, but also kind of see increase in here. And more dramatic changes will be shown in homozygous, where full size of Titin should not be there anymore. So when I ran the GILs, I did not see anything in the same area that I saw the full length of Titin. But now you can see overloaded in the the smaller size of the Titin, and also further here as well. So you can see that full length of Titin has been split into two, and then it becomes smaller. So it goes down and to the lower size of the Titin molecule. And considering the fact that I used the identical muscle that I used for the mechanical measurements, and also I used identical concentration of the protease to mimic the mechanical measurements. So this means that I didn't see the first drop, but I obviously cleaved all Titin that I used from the muscle. So this is where I got a bit sad, but obviously I really wanted to know that I made a good job in here, but obviously this mark can be gone for now. So did I cleave Titin fully? Obviously, yes, I did. So the question is, why did I not see the whole force drop? This is one thing that I don't know yet. And this is why I brought this topic to you so that I can get some input. So actin, myosin, Titin, and would there be other structures that could potentially contribute the passive force? To my best knowledge, there's no structure that could potentially contribute the passive force. And I know that this is very early for me to say there's gotta be something. So I can't say, but the reason why I have a small feeling that there should be something other than this is because of this video that I brought. So let me just briefly explain about what's going on here. So this myofiber is prepared from homozygous, where Titin can be fully cleaved, and this is after the treatment. And I overly stretched the myofibril, and what I know this is not very good, but the myosin has, and also Z disc as well, here is a bit disorganized to my point of view. So you cannot see the A band clear edges, meaning that it becomes a bit zigzagged. So the edge becomes a bit area because it has been disorganized. So that's A band. And then this is a Z disc, which used to be a straight line, but this also becomes like a range or the dark area, also meaning that it has been disorganized as well. So that would make sense to me for now, because Titin was cleaved. There's nothing can stabilize the A band here. But as I passively stretch, come on video, yes, passively stretch, and what I kind of see is that the A band tend to find its original places within the sarcomere. And now I can relatively see that the edges of the A band becomes sharper than before. So that's where I got a bit lost, because I thought I could tighten that stabilize myosin in the middle of sarcomere. But and then the Titin GL data said I cleave the Titin, but still something is here stabilizing the whole structure. So that's one thing that I need to further figure out by doing some other testing using fluorescence, so that I can see other structures not using this mode. But that's where I want to get some input. Any ideas if you have, I would love to hear. So yeah, but conclusion is that when we cleave Titin using single myofibrils, we saw a substantial passive force drop at each sarcomere length that we measured. And also we confirmed that the previous finding that we saw bigger force drop when we cleave more Titin. So obviously Titin is important, but there's something more. So this is all I have for now. And I'd be happy to take any questions.
Floor is open for questions. Who would like to start? Rob and then Hansel.
Someone in your electrophoresis, you showed that the big piece of full length of Titin disappeared and went to 2 pieces.
Right.
When the image I got from your early thing was you were going to destroy the Titin, in which case you might have got 20 tiny little pieces, but you didn't. You got 2 pieces. So I'm wondering that, which sort of suggests, as where you put your red dot over here, that it got cleaved at that point. When you came in that last video and you came to shorter lengths, is it possible that this piece, rather than having been contracted back into the corner, was able to somehow reattach to somewhere on the myosin?
Exactly. So that's one of the feedback that I also got when I was presenting my work at different conference. So that's also possible, because even if we cleave it, it's not gone. It's still there as if rubber band cut. So the structure is there. So it might have reattached to the point that I cut, or it's also possible to attach other neighboring structure in there. So in order to answer, what I need to do is to label the end of the Titin that I cleaved and then see the myofiber under the microscope with the fluorescence. So that way I know where the point is. So that's one thing that I want to do so soon, by the end of this year.
And if I could be permitted another question since I'm here. The actin filaments you have in your drawing going up here. Now, a long time ago, we used to just draw actin and myosin, and I always thought that when you pulled it to long length, all the myosin would drop down. So what stopped that? Well, it was the Titin that stopped that. But what holds the actin to be those lovely straight lines? Is there nebulin or something else joining the 2 actin filaments, which would be therefore a piece of parallel?
To my best knowledge, still it is actin and there's no structure, but that's what I find in textbook, and textbook is not always up to date, right? The feeling that I mean, I've been asking about this question to other muscle expertise, and what they said is that potentially desmin is another structure that wraps around the actin filament, and it might be still extended and then keep connected to the other part of actin filament. So, but I'm not sure. So I wanted to look for a way of extract the desmin from the sarcomere, but it sounded a bit aggressive way because so that I can potentially damage other structures as well. So I couldn't really reach the point where I can remove the desmin potentially. So that's where I am at. So, so I was like, okay, so if it wraps actin filament, what happens if I extract actin filaments then? So that's also one thing that I could trust. I could try soon, but it's on the list. I don't know yet. You know, there is an assumption that the two components might have been. Also, the reason why I thought is that if I could go back to my graph here with the blue band, I thought, you know, that blue line is what I wanted to see, but obviously the red dot goes a bit higher than the blue band. So I thought, is there other structure all of a sudden start joining in this specific length range and then contribute the passive force? Because my result says Titin, I cleave the Titin. So I don't know, that's also, but maybe if there's a structure connecting actin filaments, my assumption is that maybe it was a slack length till reach to this point, but then by further stretch, it starts coming in. But who knows? I don't know yet.
Possible on that front.
I have a series of live questions, maybe related to that. I mean, it seems like myosin is floating in the air.
Yeah.
So do we know that there is something that is still holding the myosin in place?
That's actually one thing that I would love to see, and I did my best effort to destroy the myofibril, to be honest, because that's where I got all the interest. Titin needs to be the one holding the myosin. So what I expect to see first is not even force measurements. I wanted to see A bands flying away, but that was not what I could see at all, even though I tried higher concentration of the protease and etc. Yeah, that one I cannot really answer, but there might have another structure as well, but no.
Are there literally 4 Titin?
Full Titin? No, it's just a schematic, and then I wanted to make it simplified, but I know that the 6 Titin per half sarcomere, and then recently there's a paper that's just out that how they somehow imaged how the 6 Titin goes wrap, and then how it was wrapped in the middle of, and then so yeah, that was my understanding 6, but yeah.
I'd like to have a little better imagination of the structure because we only do it as a line. Is it a line or is it a plane that we are looking from the side, or is it like a tubular shape?
Yeah, so in reality, in a single myofiber, you have a lot going on here, and then also it's a cylindrical shape. So whole pipe is going, yeah.
Lastly,
yeah.
You said you were expecting that where previous work said Titin produce like 90% of the passive force. How do they know that?
They
can be wrong. It's just 50% maybe.
I need to go back to check the details of the methodology, but they made indirect. So they found something, and then they made an assumption to make up with 90%. I need to go back to details, but no, that was my bad. Sorry.
I think so. I have that same last question about that Hansel had. It's not 90%, you know. But also in regards to sort of 66%, 90%, and you being really, well, you being optimistic here when you saw the larger numbers. I'm thinking that those larger relative differences are right there around sarcomere length 2.4.
Yeah.
And in fact, they're actually much lower when you stretch the sarcomere beyond 3.0.
Yes.
That's when you start getting 8% differences.
Yes.
So maybe do you have an idea sort of why, or do you just think it's experimental error in the heterozygous graph, why at 3.4 it's almost, the stretch is almost as high as what it was originally?
I actually do not know about that one. I tried to figure that out, and that's why I start thinking, is there other structure just joining in and start contributing to the passive force? And as we go further, there is no relatively less contribution to Titin, but other structure contributes the most. And same as here as well. So you are right that here is where I found most of the biggest changes, relative, because the force was not able to be measured. But here is actually like 35 to 40% changes. So I don't know, that's also a good point. But one thing that I initially thought is that during the incubation, when I tried to clip Titin, I set the average sarcomere length at 2.8 microns. So there is a still overlap between myosin and actin. So even if I cleaved, there might have still attached to neighboring sarcomere because it's very compact structure, the whole myofiber. There's hundreds of myosin and hundreds of actin filaments. So it is possible. Also, I calculate the length and the distance. What's also possible is this short tail of the broken fragment can still be attached here because of this distance between actin and myosin was about 40 nanometer. And then the length of this is about 45 nanometers too. So in reality, if I set it 2.8 micron, it might have still been able to attach to other structures. So that way it contributes a passive force. So my future plan is to set the sarcomere length at way longer length, where there's more no overlap at all between actin and myosin, so minimizing the chance to accidental attachment. I've tried once, and I didn't still see complete destruction of myofibrils at all, but I saw a lot lower force that goes 0 till 3.0. So yeah.
Am I just naive in thinking that you can't? So you put it in a relaxation solution. So is there just no amount of relaxation solution that can make all the myosin and actin attachments go away?
We can make it super relaxed stage 2 by putting the myosin heads attaching to the backbone. So that way there's less chance for accidental attachment.
But when you say that you had it at 2.8 when you put in the protease, don't you then put it in a relaxation solution afterwards, and wouldn't that get rid of whatever binding you had?
That's one thing that I don't know in this phase contrast image. So that's why I think I need to do fluorescence work, because I cut it to Titin, but I'm still not sure whether the structure is attached or not. So relaxing solution, it can rinse the outside, but it doesn't go inside right away. So the attachment still could happen.
So obviously completely Titin, cut it, you're not degradating it, you're not chopping it up, but you're cutting it at the A band, I band, so the questions of whether it can reach out and act like a bolt anchor and drag other things along. Couldn't you also then implement the halo cassette, tighten that disc, and really then just cut it in both places and then just blow it away? There would be no issues of it entangling, perhaps.
So you're saying cutting both parts?
Yeah. Depending, your cartoon shows it retracting, but that's not really what Titan does at the sarcomere lengths that you have. It's not under tension. it's really quite a long
Yeah.
disorganized string, so.
Yeah.
It's not like it snaps back to the Z line and disappears, it's really just long
Yeah.
floating armlets.
Still there.
So I'm just wondering if that's possible, if that's,
Um,
or that really beyond the scope of what you can do with the halo kit.
Yeah, that's one thing that we cannot do right now. The cleavage site is only cloned into this AI junction nearby here. So the place that we can only cut is here specific. But yes, it would be amazing if I could just get rid of the whole thing by cutting the Titin nearby the Z disk, yes.
Venus, or yeah, we'll go Venus first.
Do you know about the animals, like lifespan, the size of the muscles, the cross-sectional area, and all of that? Any differences?
No, we didn't. No, we didn't observe any phenotype from the across the genotypes. So that's also where we could be more confident with the results. Yes.
I was gonna ask a stupid question, but I realized it wouldn't really matter. The question was gonna be, do we know that Titan doesn't cross the Z line? Like, do you know that it sort of stops there and then a new Titan starts on the other side? Kind of how it's drawn, or?
According to the paper that I recently read, it's out like a couple weeks ago. It's sort of starting from the Z disk here. And one thing that I only remember is how it's packed in a myosin filament, but it sounded like it doesn't go through, but it's here, Z disk, and then it starts another one to another Z disk here.
Chris and then Hansel.
So I know there's some evidence that in passive force enhancement, Titan binds to actin.
Right.
And I don't know if, I don't think anyone knows if that's calcium mediated. So if it only happens if there's calcium there. But I'm just thinking that maybe if you're cutting Titan, now that Titan can float around, you can get close to actin. Maybe it's binding where we think it might be binding with passive force enhancement.
Mm.
And so I wonder if you labeled it and then looked at where it bound and then compared that to, I think, Mike Duvall's thesis shows where it binds or where we think it might be binding. And that might be an amount. I don't know how much force that would add to it, but that might be a location where it might be binding.
Yeah, so in that case, that's also a good idea to try. One thing that I would also think is that the Titan, what I heard is a bit sticky structure. So it wouldn't, so you're saying passive force, and that's when Titan is full length and fully intact. So it could bind and interact with actin filaments. But if I cut it and if I label it, then since it is cut and it's not under control anymore, so it might also just attach randomly without any calcium at all. So I'm not sure, but that's also a good way to test for sure. Yes.
Another silly question. Is it ever possible to remove some of the passive structures by some physical means? Like if you twist it or if you stretch over some limit, do we know which structure will break first? And what happens if you stretch it too much?
I think the overstretch work was done by a team a long time ago, and then it sounded, it's very robust structure, is it not? Till we, because when I also stretched the sarcomere till 5 microns, it didn't break at all. Although I lost the elasticity of the myofibrils in that length if I leave it as for a longer time, but I don't know. Not in this level, and then the thing is I cannot twist. It only moves the Z disk direction, and in order to twist I need to make this movement, which is not possible for now. It's just
As a mechanical engineering background person, I feel you should do also the things this way, that way, this way.
in the future maybe.
All right, Tim, Rob, Venus, did you have a question? Okay, so Tim, Venus, Rob.
So someone sort of stepping back out of this prep, but still with this model. I assume this type of labeling goes throughout the mouse, all of its muscles, all of its skeletal muscles have this. Kind of thinking about, a disease model of Titin. What would happen if you had a little mouse walking on a treadmill and you inject this virus in its bloodstream and then you come back 2 hours later or 2 days later? What do you expect to see?
I expect to see animal not doing very well, and actually that's one of the project that's going on in our lab now, done by other colleagues. What they're doing is they have similar model to this one, but the animals now have the cleavage sites only in the heart, and then on the live animal, they inject the virus to the body and then let them survive for 2 weeks. And what they found now, I don't seem to remember the details results, but one thing that we could easily find was that the amount of the physical movement that the mouse run the wheel, it's significantly reduced. So as if the animal don't feel well. So that's one thing that I can say for now. But yeah, that's going on.
Cardiac? That's just
Yes.
So what would happen? What do you think would you expect something similar? Do you think their gait would be altered? Would they be very just much weaker? Would they simply stop breathing? I'm curious. You're talking about passive force, and I'm sort of alluding to active force, but I'm just curious if this would be a model of sarcomere mechanics failure in a living animal.
One thing that I found, one thing that I found is that the animal just doesn't move after the injection, and then as if the animal feeling sick and even sometimes shiver at a corner, whereas the animal that we had for sham group, they are still active even though the similar injection was made. So we are not exactly sure what the animal would do with cardiac muscle cleavage. But if it is a skeletal muscle, then I would expect to see a reduced muscle size and skeletal muscle after the injection, and changed gait pattern for sure, but also reduced amount of movement as well, if it was a skeletal muscle.
If I could follow it up.
Yep.
Because Hank Rosier says that, of course, Titin turns over very rapidly. Titin is replaced in a few days. With this, if you gave it the virus and muscles, suffered this big trauma, but do you think then when the muscle is regenerated, you would restore your full Titin, even though potentially you could use it the second time?
Yeah, that could be also possible. So for now, what we are focusing on to do is to make sure it is cleaved. So we actually overdose the protease to the body, so that make sure that we cleaved. But that could also be possible that gets recovered unless there's another set of protease that goes into the body. Yes.
I'm wondering about the heterozygous model, and I'm wondering, like, you're degrading or cleaving 50%, but initially did you have, like, because we know it sticks on one side, 6 sides on the other side. So do you know that you had the marker like at 3 on this side and 3 on this side, and then when you cleave, you know that it is cleaving 3, and the same thing goes also between sarcomeres?
That much of the work, it was not done at all. One thing that I know is more about overall numbers to count the intense. So they labeled with the golden something, and then they labeled it and then see how the intensity goes up. So it's overall, not counting the numbers of it. So we are not sure about that. And 50% of Titin, it's 50% of total amount. Yeah, so it's about overall, not specific in sarcomere.
And like the other thing I noticed on the gels, I'm not sure if you've noticed that as well, is that there are 2 Titin isoforms. one is pretty strong, and then there is one above it that is very, very light. So is this Titin isoform dependent, the cleavage, like the adding the marker, the labeling, and the cleavage? Is this Titin isoform dependent specific, or it's for all Titin?
It's for all Titan, I
For all
believe.
Titan.
Yes.
Yeah, in the first, but in the second, like in the heterozygous, yeah, that's a strong one. And then on top, there is a very weak one, but I think with a stronger, like a more maybe silver staining or something, you will see that there are two isoforms.
That's one thing that I briefly talked to my boss as well. That mice, psoas muscle, there are two types of Titin in skeletal muscle that I need to go back to the details yet. But when I checked the paper, it sounded like the range of this one was skeletal muscle Titin. So, okay, but yeah, that's the doubled band that I also found in other genes too.
But it doesn't matter, like you're having, like for all Titin isoforms, you have the marker.
Yes, that's my understanding for now.
So when you had the homozygous situation, you said you expected the pieces to float away, which is reasonable. It didn't float away. You didn't get 100% drop in tension. And the discussion here is about actin, myosin, and Titin. Can this be interpreted, in fact, differently, that this is evidence for another element that we don't know about? It's a Dunning-Kruger problem, that there's a bit of knowledge over here we don't even know, except that you just have a piece of evidence that might say, let's look over here, there might be something else. And that, I don't know if you can do pico high pressure liquid chromatography where you could get these fibers and macerate them and look for some other element that's on your scans there, that's half a meter further down, something else. Is it not? Couldn't it be interpreted as evidence to look for something else we don't know?
Actually, that's also one way that I wanted to go, but I just wanted to stay as a bit conservative and safe as well, because that's not what's out in the literature. But yeah, that's one way to prove it too.
Because it's sort of evidence that there's another element.
Could be also true. Yeah.
By the way, Rob, down there are lots of proteins. On this one and that one, and there are lots of.
Well, that's more supporting evidence.
Yeah. So just to give you an idea of the size, this is a 3-point megadalton, and then this is really big. Most of the protein that's analyzed primarily, it's thousands of times smaller. So it goes kilodalton units.
There you go, lots of options.
So yeah, it can go like 10 meters down, Jills maybe. But yeah, that's one thing to do. But also one thing that I am not fully understanding, and then maybe Venus, you could correct me, but in this bigger level, there's no letter standard. So even if we find something on the Jills, then we need to go back to literature to check what is this band could potentially mean. So there's something unknown. Things can still be shown, but we do not know what that is.
Yeah, because if you know that they're down there, then exactly that. What are they doing? They're going to be doing there for some purpose, and one of them might be contributing there.
I wanted to add on to that if you look at the results, it almost seems like there is some sort of a backup mechanism. Maybe Titan was doing 90% under normal conditions, another one was doing most of the time, and then starts to continue when something's not working.
Yeah.
Because it's really weird that acid push sort of recovers.
More evidence of another element.
Right.
Yeah.
So these were disseminated
Yes,
samples, not
Yes.
frozen.
yes.
Why there are two bands? Is this the two pieces? But they are
Here,
the two pieces of Titan, one piece on one side and one piece on the other.
Yes.
That's two
exactly. So we cleave one spot, and so we get now two parts of the Titan. One is related to I-band region that's known to be here somewhere, which is 1.4 megadalton, and the other part is a little longer. It goes up to 2.2 megadalton. The part that goes into the myosin.
But why there are two bands then at about 2.3? Like,
I'm sorry. So maybe it should be easier for me to just bring this one. So the broken part from the I-band region goes here, which is this one. But the part here would known to be go here. So there are two parts after cleavage.
so
Yeah, go
I
ahead.
have
Venus.
questions. So how did we quantify the 46 and 97?
Yeah, so
98%?
So there's a software that we use. We run, it's called a multi-gauge. And I cut it in here, but I have actually a lot longer data bands that I can use it for normalizing it. And then even though I calculate the protein amount for each lane, but there's all human work, so there's a bit of error. So I always need to normalize it. So if I say this band, but then I need to know how much the total protein that I put in this lane. So a lot down there, there is a value that I can normalize it. So that's the value relative to this one. And then that's value relative to the other one. And these are usually representing the total amount of protein. So.
Yep, yeah, of course.
Yeah, the other more like question about the experiments and the past course is that, do you know if you have one myofibril in your that was tested, one myofibril that was tested or more? Because if you have in parallel, we know that desmin it might play a better role by connecting the myofibrils in parallel and stabilizing the myofibrils. Not necessarily the A band, but just the myofibril, just in parallel would stabilize them.
Yeah.
So and the myofibril that you showed, it seems thick to me.
Yeah.
So
yeah, that's also true. Before I do the measurements, I just to apply retention just to try to see if there's any possible separation if there are multiple myofibrils. And so that's why I sometimes try to avoid to use the multiple myofibrils bundle of it. But it's also possible that the myofibrils attached to the other side that I cannot see. So it might have looked like one here, but could be here too. So actually that's also possible. So that way we have more contribution from the desmin part.
Is any other questions for someone? Okay, none?
What-
Oh, yeah.
why did
Venus?
you start the
Absolutely.
sarcoma length of 2.0? Because is it the resting length in like in mouse myofibril?
Yeah, I wanted to just use it as a forces value 0 and then goes to.
But does it shorten this much? It's not slack?
It's a slack. Yeah. So it's average sarcomere length, assuming that it's there. So I have a 7 sarcomeres in a row, and then I measure the average length. So that's why its nominal length is 2.0, but it doesn't actually go down there. Yeah.
I have one final question, someone. In
And
the
Tim,
video,
yeah.
at the end, when you bring it all back together, it looks like kind of like some of Chris's videos where it goes slack and then it like recovers a little bit.
Yeah.
Why is that happening? What's responsible?
I, to be honest, do not know what it recovers. So that's also where I start thinking there might have been some other passive elements contributing to the passive force, because in that video, Titin is gone, so you shouldn't be recovering and then recoiling back, but still it goes back to original length. So that's why, okay, so there's that other passive structure, but I don't know, to be honest, I don't have a good answer for that.
In the video, it looks like you don't have homogeneous disruption across the entire myofibril. Like the biggest disruption is in the middle, but but I thought with the myofibrils generally you got the same. The idea was that the strain is going to be constant across all of the bands.
So I think that happens depending on the myofibril. If they have a specifically weaker sarcomeres in the middle, then they tend to stretch a little longer. Also, it depends on how I pick the myofibril as well, because sometimes most of the time I see the damage at the edges. So that depends on the myofibril, to my opinion.
Tim.
So just to follow up on Venus's question in your response, that you're really cleaning all of things, you're adding the protease at the sarcomer 2.0, which is very short. But why, don't want to make work for you, but why don't you stretch the myofibril to say 3.2 or 3.3, hold it, then infuse the protease? You'll watch, hopefully you'll watch the pores disappear. And if there's any, if any of these arms are all flapping around trying to attach to something, maybe you can't because the sarcomer is so long.
So your question is, why did it not?
Yeah, why did you considering not doing a series of little stretches like you've done, but really just stretch it out to a long, like hold it till it's there in steady state, and then add protease as soon as the cleavage occurs, the force will draw back. And if there's the argument here about whether or not these broken bits actually can be sticky, well then it won't really matter because there's no way they can reach out and grab something because the myofibril will be longer. I'm just curious if that would be a parallel set of experiments.
Yeah, so that's actually one way that I'm thinking to do, to put the sarcomere really long length and then actually record for a 10 minutes video during the incubation and then see how the force drops. Exactly. And the reason why I didn't do it first is I wanted to confirm the previous findings, so that way I did exactly the same experiments compared to that, but only difference is the structural level that I tested it. So as soon as I'm done with the data collection for this one, that's one way, one of the many experiments that I'm thinking to do. Yeah, I like your idea, Titin.
What happens if you activate? What's your
Um,
expectation?
No, I actually have done activation. So the disorganization of the structure becomes a lot worse. Z disc, sorry, A band goes either side of the Z disc, and as I activate it and leave it for a longer period, then I cannot say which one is Z disc and which one is A band at all anymore. It becomes all masked things up as if myosin goes one direction all the way to the end of the sarcomere, and then the below myosin goes to the random direction also. And then I tried to deactivate it as well, and then that structure, once it's mangled, it couldn't be recovered.
That's another proof that Titin is not there anymore.
True, exactly. But yeah, I saw a lot of ugly images from my experiments.
Awesome. Any other final questions, comments for Seong? All right, let's thank our speaker again.
Thank you.